Hello, this is Ed Chapman, and this video cast is going to be covering the structure and function of proteins, which are a group of macromolecules made up of monomers. Alright, the building blocks of all proteins are called amino acids. So amino acids are what we call the monomers of proteins. And there is a set of 20 different amino acids that all proteins on Earth are built from. And we get these amino acids from the plants we consume or from the animals that we consume that in turn must have consumed a plant at some point in the food chain. And this picture here shows you guys all the different amino acids that are used to build all the different proteins. And you guys are not responsible for knowing any of these structures except for the commonality. What do they all have in common? And if you look closely, for example, this amino acid is called glycine up here. You notice it has an N hooked to a C, hooked to a C. If we go here over to alanine, it has an N hooked to a carbon, hooked to a carbon. Here's valine. It's N hooked to a C, hooked to a C. So all amino acids have a nitrogen hooked to a carbon, all in a row. And this is the hallmark of all amino acids. Everything else about the amino acid is the same except for what you attach to the central carbon. So for example, in glycine, glycine has a little hydrogen attached. Alanine has a CH3 attached. Valine has a much more complicated set of carbon attachments here. Leucine's getting even bigger, all right? But these yellow or orange shaded parts show you how the amino acids are different, but they all have in common this basic NCC structure. So let me show you guys a little bit more closely what we're talking about here. So all amino acids share this basic structure: a nitrogen hooked to a carbon, hooked to a carbon, with something else attached to it. All right, I think that's as deep as we need to go into the structure of an amino acid for you guys to be able to pick it out of a lineup of pictures. Now, to build a protein, all you have to do is hook together amino acids in the right order. All right, and you do this by forming peptide bonds. So if we attach, for example, amino acid number 1 to amino acid number 2, we build these peptide bonds, which are a special type of covalent bond that's holding the amino acids together. And because they hold amino acids together in a protein, okay, we call it a peptide bond. So remember, protein, peptide, those go together. So this could be the amino acid alanine, or this could be valine, or leucine, or what's another one? We could do tryptophan. There's 20 different ones, but it could be alanine again, although that wouldn't be right because we've got squares representing alanine. So this is a circle, so this one would have to be valine again. So the idea here is you hook amino acids together by using peptide bonds in a chain, and this produces what we call polypeptides or proteins. All right? So a polypeptide is the same thing as a protein. Now, most proteins found in animals and plants are hundreds, if not thousands, of amino acids long. So they're very complicated, very large molecules. And one thing that happens to a polypeptide chain that's very important is the chain doesn't stay straight. It gets folded up. Okay, so the idea here is you start out with a chain of amino acids, okay, all sorts of different shapes, and then they get all kind of folded and wrapped and spiraled up into a glob or into something more complicated to give it a three-dimensional shape. And it's the 3D shape of the polypeptide that gives it its function or allows it to do its job. All right, here is a computer animated or computer drawn representation of a very common protein called hemoglobin. And you can see that this hemoglobin molecule has areas of it where the polypeptide chain is kind of straight. You have places where it's been spiraled up like a spring, like here and here. You have places where it's connecting. All right, it's a very complicated molecule. And this three-dimensional shape is what allows a protein like hemoglobin to do its job. And in the case of hemoglobin, it carries oxygen. It's what makes your blood red. All right, so what I'm hoping you guys can now see is that the shape or conformation of a protein, okay, conformation means the same thing as shape, okay, determines what it can do. So some proteins have a conformation or a shape that makes them very good at building stuff, okay? So, for example, here's a very common protein found in humans. It has a three-dimensional shape that's very long and a very strandy shaped. This is a protein called keratin, which is used to build hair and fingernails. Okay, hair and fingernails are things that grow. They have an older part and a younger part, and they have to be flexible and waterproof. These are what we call structural proteins. They're building body parts. Another one is called collagen, which performs a similar role deep in your skin. So keratin and collagen are two structural proteins because their shape allows them to do their job, which is to build hair, fingernails, and components of your skin. Okay, other proteins have a conformation or shape that allows them to do things rather than build things. And these are called functional proteins, all right? Now, functional proteins do a job. Kind of like a bucket's job is to carry something or a rubber band's job is to be stretchy, there are proteins that have shapes that allow them to do jobs. For example, hemoglobin is a protein that has a shape kind of like you can think of it as analogous to a bucket that allows it to carry oxygen. Actin and myosin are two proteins found in your muscles, and they can expand and contract to allow you to move, kind of like rubber bands. So again, the conformation or shape of a protein allows it to do its job, and there are basically two kinds of proteins: structural proteins, which build things, and functional proteins, which perform jobs. All right? Now, this introduces a really basic concept in all of biology. The structure of a thing determines the function of a thing. So how something is built is going to give you a clue about what it's going to do. All right? And this is incredibly important in proteins. The structure, or the three-dimensional shape, okay? The structure of proteins, remember, is a three-dimensional shape, allows them to do their function. And there's two functions for proteins. There are the structural proteins, which build body parts like hair and fingernails, and then there are the functional proteins. Okay, and these are proteins that perform jobs like hemoglobin and actin and myosin. Now, because proteins have such a complicated three-dimensional shape, anything that changes this shape will make the protein no longer function properly. So, for example, in this picture, excuse me, we have a normal protein with its normal three-dimensional shape. This could be something like hemoglobin, and if we denature it, okay, which means that we change it, its shape changes. So now you can see this protein is kind of unraveled quite a bit. This is what we call a denatured protein. And there are three different ways that I want you guys to be aware of that you can denature a protein or disrupt its three-dimensional shape. You can change the pH, especially by putting it in an acid. You can increase the temperature, which is basically you can cook it, all right? Or you can mix it with different solutes, like salts. Okay, so if you make the environment that the protein's in very salty or very hot or very acidic, you can change the shape of it, and that's called denaturation. Excuse me, getting over this cold. You've seen this, everybody has witnessed this denaturation process. Excuse me, when you cook an egg, all right? This egg was dropped into a hot pan, and it looked like a raw egg, but after it was exposed to the temperature, the proteins in the egg white especially noticeably changed or denatured. Excuse me, another name for this is cooking. And I'm sure everybody knows that once you cook an egg, you can't uncook it. So the renaturation process is something that doesn't happen very often in everyday biology. Usually when you denature a protein or you cook a protein either with heat or with pH change, you change its structure in a way that it can't go back to what it was. Excuse me. So this process is called denaturation. All right, hair and muscle are built from two very different types of proteins. If all proteins are made from the same set of 20 or so amino acids, how can muscle be different from hair? Can you guys explain this? If you can, then that would convince me you understand the difference between structural and functional proteins. All right, we're going to stop there. The next video cast will pick up with nucleic acids. Thanks for listening.